now we are going down to a very small group called the vertebrates. We are vertebrates. We were among the bacteria and extraordinary things, fascinating, in the very, very deep past. But now we are just back to the at a small scale. Oh, some problem? Okay. Ah, right. Anyway, vertebrates are a relatively small group of animals which provides the advantage of having a phosphatic skeleton, bioapatite, which preserves fairly well in the fossil record. And it is usually vertebrates, except some of vertebrates which I'm going to show you soon, which are just as poorly mineralized as a slug, and when they thank you very much, Karen. Some of these early fossil vertebrates are just like squashed slugs on a highway. You know, it's completely flat. Anyway, they can provide some information, but otherwise, most other vertebrates have a very extensive, relatively extensive skeleton, be they early vertebrates or even quaternary or tertiary mammals or dinosaurs, and vertebrates are relatively complicated. So when they are fossil, when they occur as fossils, they provide a relatively large number of characters which can be used to reconstruct phylogenies. Here is the earliest vertebrate fragment ever discovered. It's about 475 million years ago. It was discovered in Australia. And why is it a piece of vertebrate? It's because, oh, there is a pointer here, perhaps. It displays, it's a small piece of bone with the tubercles or outgrowth on the surface, and it displays a groove like that, which is the sensory line groove, which is found in all fishes, practically, and it houses the neuromasts, which are sensory endings of the lateral line system, and these tubercles are made of dentine. So there are two characters here, sensory line grooves, which means that this animal had neuromasts, lateral line system, and thus that in its development it displayed neurogenic placodes, which is a unique character for the vertebrates. Then dentine tubercles, we know that there is dentine, not on that specimen because it's preserved in imprint, but in other specimens of the same, virtually the same age, the tubercles are made of dentine, which means that they were odontogenic tissues, and which usually come from neural crest cells. I say usually because it's for sure in the head, but it's not absolutely clear whether neural crests in the development, ontogenic development, if a trunk neural crest, neural crest cells of the trunk, provide or generate odontogenic tissues. So that's the introduction, this earliest piece of vertebrate. This is what I am working on. It's not very exciting, but sometimes you will see there are very beautiful specimens. Let's go back to the structure of the group, of the vertebrates. Here is a vertebrate phylogeny. On one side are the gene, it's a tree based on gene sequence data. On the other side, it's a tree based on morphological and physiological characters. They are fairly well resolved, and there are some inconsistencies, but it's not very important. And in fact, some recent molecular trees provide a much better fit with the the morphological and physiological, phenotypic data. The vertebrates comprise a small group, which is now proved to be monophyletic on the basis of gene sequence data, but it has long been thought that it was paraphyletic. Anyway, it's called the cyclostomes. They are jawless fishes with lampreys and hagfishes. And all the rest of the vertebrates are jawed vertebrates. It starts from sharks to tetrapods. That's we are tetrapods, four-limbed vertebrates. And between these two organisms, there is a long segment of the phylogeny of the vertebrates. Here again, there is quite a good fit between the molecular and phenotypic data, except for some questions, but it's not very important. But, sorry, if we only had this, remember that these are the extant vertebrates, the living vertebrates. And if we only had these two big groups, the cyclostomes on one side and the gnatostomes or jawed vertebrates on the other side, how could we imagine the transition from the evolutionary transition and the morphological transition between jawless vertebrates and jawed vertebrates? Unfortunately, in the past, in the Paleozoic, and since the 19th century, we know that there were fishes, very bizarre fishes that looked sort of, some of them like these ones have a whole sort of a horseshoe-shaped skull. And for a long time, nobody knew exactly what they were. Some people thought they were catfishes because they vaguely looked like catfishes or sturgeons. But in the beginning of the 20th century, the famous paleontologist Eric Stensio decided to tackle extensively, to study extensively these fishes, which were collectively called ostracoderms. We didn't know exactly what they were, but they got a name, ostracoderms. And in fact, ostracoderms means that they are, we know, soon after, after the works of Stensio, it was demonstrated that they were jawless fishes, and but heavily armored and heavily ossified jawless fishes. So, and these ostracoderms range from about 470 million years ago and up to 360 million years ago. And then they all became extinct. Stensio, when he realized that the anatomy of these ostracoderms was more or less similar in many respects, although the overall aspects of these ostracoderms is quite different from lampreys. But there are some anatomical characters that was described by Stensio, which recalled the anatomy of lampreys in some respects. And Stensio in 1927 suggested that these ostracoderms, these armored jawless vertebrates, were ancestors of the cyclostomes, of lampreys and hagfishes. So he supposed, he called that the diphyletic origin of the cyclostomes. In fact, he thought that the skeleton had been lost at one time, separately, twice separately, in hagfishes here and in lampreys here. This idea is now completely abandoned, and during the 20th century, many new ostracoderm groups have been discovered. Here in pink, you have all the ostracoderms that you can find in a typical ostracoderm fauna of the 400 million years old, for instance, in Spitsbergen. And these ostracoderms lived alongside the earliest jawed vertebrates, such as this precursor of lungfishes, and these armored, the placoderms, these armored jawed fishes. And you can see that these ostracoderms have a wide range of different morphologies. Some have paired fins, the first paired fins. Others have no paired fins, they just look like a box. And now, after many years of study of ostracoderms, we came now to this topology, this tree, which shows that all ostracoderms are in fact a paraphyletic group. They form a paraphyletic ensemble, some of which are the sister group to jawed vertebrates here, the osteostracans, for instance, and others are very far, much lower in the tree. And all these other groups are devoid of mineralized tissues, such as the hagfishes and lampreys. There is absolutely no bone, no teeth, nothing, no mineralized tissues in cyclostomes. So, in a sense, you can see that all these ostracoderms, which are restricted to the lower Paleozoic and middle Paleozoic here, are completely extinct, and they form a sort of they document the progressive assembly of the anatomy of jawed vertebrates. In fact, they are precursors of the jawed vertebrates, although they had absolutely no jaws. Here is exactly the same tree, or almost so, in a more geology-friendly form, because the previous tree was a tree for zoologists or for biologists, and this is a tree for geologists. But it's the same. And you can see that all these ostracoderms are jawless, simply jawless stemmed gnatostomes, with osteostracans here as sister group to the jawed vertebrates. And there is a big segment of this of the vertebrate tree, which in fact is totally devoid of mineralization, mineralized tissues. So I'm not going to bother you in detail with all these ostracoderms, but the most interesting in the present connection are the pterospidomorphs, which contain three major groups: arandaspids, astraspids, and heterostracans. And these pterospidomorphs are among the earliest skeletonized vertebrates. Unfortunately, here is one of these, the earliest Ordovician arandaspids, such as Sacabambaspis, which is very bizarre. It looks like a tadpole, or my students say it looks like a suppository. But anyway, it's fairly well documented now. This specimen is a good specimen, but we have now even more complete specimens, and now we are able to reconstruct extensively the shape of these animals. But there is nothing inside it, inside them. No preserved structure, no cartilage. Well, there was probably cartilage, but it doesn't get preserved. And the exoskeleton, all the skeleton, all the mineralized parts are belong to the exoskeleton, that is the superficial skeleton of the animal. Sometimes in some other pterospidomorphs, but much younger ones, such as heterostracans, you can find impressions of the internal organs on the surface, on the ventral surface of the carapace, which covers the head, where you can see traces of the gills, traces of the semicircular canals, and the brain, and the eyes here, and the olfactory organs. So, but this is very poor information, in fact. And with other ostracoderms, such as the Galeaspids and osteostracans, at last we get information about the internal structure, because the endoskeleton of these fishes was slightly calcified. There was a slight perichondral bone lamella on the surface of the internal cartilages, and Galeaspids and osteostracans, which are here, are now fairly well known. Here is a Galeaspid, for instance. This is the external shape. It's an animal with a big opening at the anterior end of the carapace of the head, which probably served to inhalate the water, the respiratory water. But some others, in fact, some galliaspids show the internal structure. Here you can see the lateral head vein, the brain cavity, or the cavity that contains the brain, and the semicircular canals, and very numerous gill pouches, or at least the fossae, the depressions that house the gill pouches. Certain galliaspids have up to 50 pairs of gill pouches, which is unbelievable for most zoologists, and especially evolutionary developmental biologists, who say no, it's absolutely impossible. No, we have these fishes, and they are preserved. So we have up to 50 pairs of gill pouches in galliaspids, and that's all. It's not the first time that polybranchial, such a much enlarged number of gills occur in the vertebrates anyway, but these ones are very spectacular. In osteostracans, here we have an example of a small skull of an osteostracan. The animal looked like that, with small paired fins on the side. And this specimen has been prepared, it's seen in ventral view, and it has been prepared with very weak formic acid to expose all the canals which house the nerves, the brain, the orbital, the eyes, etc. And you can see that all these canals are in fact formed by perichondral bone, very thin layer of perichondral bone, which is makes them be very fragile. And now, thanks to the use of synchrotron CT scanning, we can have very good resolution of this, and very good images of these internal structures of these early fishes, these early jawless fishes. So, vertebrates have two skeletons. One is the endoskeleton, which you have just seen in an osteostracan. It's preformed. The endoskeleton is always preformed in cartilage, and then which subsequently can become ossified. The exoskeleton is a superficial skeleton, which is called also the dermoskeleton or dermal skeleton, and it is not preformed in cartilage. This is the superficial bone of all these fishes I've just showed you. When you see these very bizarre shapes, external structure of the exoskeleton with plenty of small tubercles, it's only the exoskeleton. We have exoskeleton, but it has been it's sunken into beneath the skin, and but we are not going to speak about this late occurrence of the dermal skeleton. So, coming back to the endoskeleton, this is a long story made simple, short. It starts with cartilage. It subsequently becomes covered with perichondral bone here, and sometimes there are some internal calcification of globular calcified cartilage inside. And then there are two possible ways. Either the perichondral bone is lost, except that sometimes it remains in form of small patches, and replaced by prismatic calcified cartilage. And this is unique to, the prismatic calcified cartilage is unique to sharks. It's only found in sharks. Another path is when the perichondral bone remains, and then the cartilage is invaded by endochondral bone. And this is what happens in our bones. Spongious bone is endochondral bone. But in most of these early fishes, in the lower Paleozoic and middle Paleozoic, as I told you, there is only perichondral bone, perichondral lamina, which a thin lamina which lines the internal canals and cavities for the internal organs. But sometimes in placoderms, which is an extinct group of jawed vertebrates, the sister group to all other jawed vertebrates, probably paraphyletic by the way, And here is the skull of a placoderm. Here is a skull of a placoderm which has been etched with, slightly etched with formic acid, with a weak acid. You see the brain cavity here, the cranial nerves, the cavity for the otic capsules, and all of these structures are lined with a very thin layer, a paper-thin layer of perichondral bone. And that's all we can find, we can expect to find in both ostracoderms and placoderms. Calcified cartilage, as I told you, it occurs, it's unique to, prismatic calcified cartilage is unique to sharks. This is a gill arch of a modern shark with all these small prisms of apatite, of calcified cartilage. And it exists since at least the Devonian, in 400 million years ago. This is the ventral side of the brain case of one of the earliest known sharks, and you can see all these small prisms exactly the same. So it's unique to chondrichthyans, to sharks and allies. The exoskeleton is extensively developed for instance, in placoderm, such as this artodire from the Devonian of Australia. It's covered with small, it's bone, but it's covered with very small tubercles, which in fact are, the structure of which is the same as our teeth. It's dentine. And the odontodes initially are found as separate scales, like in the scale of a shark, for instance, but in most of these early gnatostomes and also in ostracoderms, the odontodes are attached to, basally attached to the dermal bone, to the superficial bone, and this provides sometimes information about the history of the patterning of the exoskeleton. These small tubercles, as I told you, they display virtually the structure of our teeth, but there are different kinds of dentine. And these are just descriptive terms, mesodentine, semidentine, metadentine, orthodentine. These are merely descriptive terms. It doesn't show us the polarity of the evolution of these, of the dentine, except that some types of dentine, such as the mesodentine, has been thought to be intermediate between bone and dentine, but it's not sure. This is an example of mesodentine in a Silurian osteostracan, a jawless vertebrate. You can see that all the cell spaces are connected by cell processes, and at last, near the upper part of more superficially, the odontocytes or odontoblasts are grouped in small spaces, which more or less suggests that it's comparable to what we call the pulp cavity in more advanced teeth. Semidentine is another type. Is another type of dentine which is found only in placoderms, such as this little Devonian placoderm here. You can see there are tubercles. If you cut the tubercles, you can find semidentine, and semidentine is unique to placoderms. It doesn't exist any longer in present-day nature, and it shows small cell spaces for the odontocytes, which are with a single cell process, which can sometimes eventually branch. We don't know exactly what's the meaning of semidentine. All what we know is that it is unique to the placoderms, to armored jawed vertebrates. And the most impressive state of the dermal skeleton is represented by cosmine. Cosmine is a very strange, it's exactly the same as the structure of the teeth, but it forms a single layer which entirely covers the bones, the dermal bones of the head, for instance, of this sarcopterygian fish. And it's not only covered, this tissue, hard tissue, not only covers entirely the bone, but it is also covered by enamel, true enamel, which means that the animal could not there was no skin respirations anymore, and the only contact between the outer environment and the nervous system of the fish went through all these small pores which are covering the head here and there. These are an example of this is an example of the amazing diversity of the hard tissues in these fishes. Now I'm going to speak especially about this period, which is the lower Ordovician, lower Ordovician, about 450 million years ago and later, down to this the upper Cambrian. This is the period when vertebrates, the first vertebrate skeleton appears, and before that we have nothing for sure. Here is, for instance, an example of a late Ordovician scale. Perhaps it is a scale. We don't know what it is. It's most of these remains, except for a couple of entirely preserved, completely preserved fishes, specimens. Most of these hard tissues are represented by isolated scales, which are found by macerating rocks in formic acid or in acetic acid. And look at this little scale, which is about 1 millimeter in diameter. It's called an odontocoplex, which is a grouping of small odontodes. This is a basal view here. You can see the pulp canals and the odontodes, which are being added to the lateral side of the scale. And strangely, a section of these kind of scales, such as Tesacophiaspis, for instance, shows a sort of, there is no dentine. It's totally acellular. It looks like dentine. There is a pulp cavity here, but it's completely acellular. And my colleague Valia Karatayutetalima suggested the name lamelline for this tissue. We don't know what it is. There is no equivalent in the present day nature, except that some living teleosts show on this, namely catfishes, show sometimes such odontodes which look like lamelline in structure, but it's not given. It's not sure that it is true lamelline. Other of these scales from the Ordovician of essentially Central Asia show exactly the same base of a base bearing odontodes of lamelline, which are grouped, very numerous odontodes, which are grouped to form a scale which superficially resemble a placoid scale of a shark. So these scales have been referred to possibly shark-like creatures, but we don't know. Among the other Ordovician fishes, which are the famous, here is the famous Astraspis from the Harding sandstone. Here we are in the Darreauvillian, the middle Ordovician. And it's one of the few specimens which provide information about the entire dermal skeleton, but that's all the kind of the information we can get. Sometimes there are isolated scales that are made of mesodentine plus a little cap of enameloid, and you can see these the cell spaces in these in these scales. We don't know what this form is. It has been called sciictis. We have no idea of what it is. Maybe it is a placoderm already in the Ordovician, but it would be far before the first placoderms appeared or were recognized. And also, along with astraspids and in the Middle Ordovician, still in the Middle Ordovician, there are a lot of scales which have been tentatively referred to thelodonts, that is a group of ostracoderm with a micromeric skeleton with isolated scales, and possible chondrichthyan scales, but it's not very clear. It's not sure that they are actually chondrichthyans. These are arandaspids. These are the first arandaspids ever found from the Ordovician of Australia, a little bit older than the Harding sandstone material. And along with arandaspids in Australia occur also these very bizarre scales, which are empty. Apparently, there is no dentine. It's just like a superficial coating. It looks like shark teeth, more or less, but personally, I'm not sure that this is vertebrate. It could possibly be some kind of arthropod. We don't know. The structure, the histological structure is very poorly preserved and poorly visible. Our friend Sacabambaspis from the lower middle Ordovician. Here is the image I showed you a bit earlier, and the structure clearly shows tubercles. The dermal skeleton, the exoskeleton clearly shows tubercles, which are composed of dentine with a very very thin cap of enameloid, and below that there are big cavities. Apparently, this exoskeleton was relatively light. So this is the first pictures I showed you, the earliest vertebrate remain, which probably belonged to an animal called Porophoraspis, which probably looked more or less like the previous, like Sacabambaspis. And now, knowing that, some of my colleagues have been desperately trying to find evidence for bone or for even fragments of bones in earlier and earlier layers down to the Cambrian, and they found some bits and pieces which have been referred to as anatolepis. What is anatolepis? We don't know. In fact, the structure vaguely shows to be canaliculi, which are more or less suggestive of dentine. That's why my colleagues, my British colleagues, suggested that it was a form of a precursor of dentine. I'm not convinced, and also again, anatolepis doesn't look very fishy. It's probably some kind of carapace of an arthropod, but which arthropod we don't know. So that's all for the bone. And do I have time? 10 minutes. 5 minutes. Okay, so that's 5 minutes. Okay. And before bone and dentine, as I told you, there is a big segment of the long segment of the vertebrate phylogeny, which is populated by animals that are not preserved, that did not produce bone, neither bone nor dentine. Among them are some fossil lampreys and presumed fossil hagfishes. And here is an example of from the Cambrian of these fishes that look like squashed slugs, as I say. They are just imprints in shales, but nevertheless you can see traces of the myomeres, the muscle blocks, the eyes, the olfactory organ here, and this is probably what Hycoictis, these miloconmyids, look like, just like an amphioxus with a head, more or less. Another one was found very recently in the Burgess shale, and it's also a squashed slug, completely crushed, but better preserved than the Chinese ones, in the sense that you can vaguely see traces of gill arches, which are transformed. It was cartilage, but now, as it is, it has been completely epigenized in pyrite. And you can see here the eyeballs here, and this is more or less what the animal looked like. It's horrible. It's like a naked children's safe version of Sacabambaspis, perhaps, with no bones, like a Sacabambaspis without bone, and much smaller indeed. But that's all what we have in the Cambrian. And also we have lampreys, but much younger layers. In the Carboniferous, for instance, we have good lampreys, squashed slugs again. But anyway, they show some traces of the eyes and gill pouches. And conodonts. Kevin mentioned the conodonts. Conodonts are small denticle-like fossils, which are mineralized. But it's the only part of the animal that is mineralized. And they look like teeth. They have been vaguely interpreted like teeth, and that's how we can reconstruct a conodont. They look vicious, but and why we can reconstruct the body of the conodonts? It's because the conodonts have been found in sometimes they are very rare, but exceptional preservations of the soft tissues. Here is the tail of the conodonts, which show the fin radials. Here are the eyes. These are the myomeres, the muscle blocks. It's not much. But anyway, strangely, for about 10 years, my English colleagues have been very active in defending the theory that conodonts are vertebrates. And in fact, because of this similarity in structure, this bioapatite composition of the conodont elements. And recently, there have been a lot of struggles about the affinities of conodonts. I've never been convinced that the conodont denticles were actually homologous to the odontodes, the scales or the teeth of vertebrates. The growth is, the mode of growth is totally different. And recently, last year, my English colleagues went to Canossa, as we say in French. They studied the conodont elements at a high resolution with a synchrotron microtomography, and they showed that the growth of the conodonts is totally different from the growth of the mode of growth of odontodes. And in fact, the growth of the conodonts is just like small glasses that you would pile starting from the top and then growing basally. And it has nothing to do with what we have in vertebrates. Plus the fact that the enameloid or enamel-like cap was also growing basally, especially in the most advanced conodonts, the euconodonts. So exit the conodonts, except that they are probably vertebrates anyway. But their teeth or the denticles are not homologous to the vertebrate teeth. And to end with that, some years ago, I was studying again squashed slugs, a fish from the Devonian about 370 million years ago, old, a fish that is preserved as an imprint with a very bizarre, well, it looks like an anaspid. This is the tail, the head, the eyes are somewhere here. But this Euphaneuripids possess a long series of gill arches. Nobody believed me when I said that they were gill arches, because it's impossible. As I told you, the developmental biologists, they say, no, it's not possible to have that many gill arches, because there would be 30 pairs of gill arches extending to the anal fin here. And then I came across some very large specimens of Euphanerops with the gill arches in which the gill arches became calcified. In fact, they were cartilage, the gill arches were supported by cartilage, and they started to transform or to generate globules of calcified cartilage. And this is what they look like. They look like double, paired structures with two cavities for the chondrocytes, and which turned out to be exactly similar to the chondrocytes of a lamprey. Here is a chondrocyte of a lamprey. It's a very special structure. In lampreys, the cartilage has a very special structure. Although it doesn't, it is never mineralized, but here in Euphanerops, it was mineralized. So it's the first possibility of having at least some calcification in fishes which were thought to be absolutely devoid of any mineralized tissue, and it provides some information about the internal structure. Thank you very much.